11:30, 1 hour.
இது அந்த இதுல இருந்து கொஞ்சம் சொல்ல முடியுமா? பாதி போட்டு சரி ஓகே good morning. We have to finish. We were finishing up the cardiac cycle ECG. And now today we have to finish 3 things, that is blood grouping, coagulation, then control of cardiac control of cardiovascular disorder. And in that you should know the another one, transport of gases by the blood. That along with the functions of the blood, transport of gases by blood. And these are the things which you are supposed to complete today. Now the blood grouping, the first one is the blood grouping. Now the blood grouping, grouping of the blood, that is blood group. There are how the blood is grouped according to, there are 2 substances in the blood called antigen. What is antigen? It's a layer of a protein layer on RBC. Antigen is a protein coat, a protein layer on RBC, on RBCs. Now this protein layer, these are 2 types of protein layer, that is type A and B, 2 kinds of it. Then in this, according to the antigen, the blood is grouped as A antigen and B antigen, that the A group and B group. Some of the RBCs will have only A, then you call that as a A group. That is not, say, average. Now RBC count is in million, 4.5 to 6 million. So if 90% of the RBC has got the A antigen, you call that as A group. Similarly, 90% of them will have the B group, then it is B antigen, it's called B group. Some of them will have both antigens will be together, then AB group. Some of them will have no antigen, then you call it as a O group. This you already know. And the blood grouping, the grouping is done by the scientist Landsteiner. Now the antigen will produce in your body antigen. Antigen is nothing but a protein similar to WBC. It is phagocytic and it helps in the defense mechanism of the body, protecting the body against the infection. But the antigen and antibody are opposite to each other. Antigen and antibody is opposite. Antibody is the one which will be produced by the antigen. So anything, there are 2 types of antigens in our blood. Water antigens, they are protein layer on the RBC, and there are 2 types of them, A and B. Majority of the RBCs has got A antigen, A coating, it's called A group, the blood group, B antigen, B group, AB group, and O group. These antigens will have a tendency to form antibody. What are antibody? They are WBC-like cells. In a sense, they are phagocytic. Phagocytic. They swallow the foreign bodies and produce immunity, resistance to disease. Immunity. So now that is according to the tabular column. Now this method of grouping it's done by Landsteiner. It is called ABO grouping. ABO group system. You want the A group, B group. It's called the method of grouping is known as the ABO system. Under the ABO system, you have like this way. You have antigen. Now the antigen and the antibody are opposite each other. You know the A antigen, B antigen. Suppose you have got antigen here. Antigen A, B, C, A, B, and O. Then the blood group. The blood group is known as the A group, B group, AB group, and O group. But antigen will be antibody will be opposite. With A group, A antigen will produce B antibody, B antigen will produce A antibody, AB will produce no antibody. Oh, the no antigen means AB, both the antibodies will be produced. So the antibodies and blood group will be opposite. Now, what is the use of antibody? It provides resistance. There is a rare group which is also known as ABO group. ABO grouping. In this kind of method of grouping is called ABO grouping, done by Landsteiner. And why should you know what is an antibody? What is an antibody? It produces resistance. So when if there is if two different antibodies come together, now in the case of AB group, it's naturally present antigen. O group AB antibody is there, that percentage will be different. Suppose it's a purely is A group which has got maximum B antibody, administer B blood. Now the two different categories artificially mixing up will club up the blood cells. It will be fatal. It will gather up the blood cells, group the blood cells together, and the blood cannot work. The normal function of the blood cannot be carried out. It may be very fatal, and suddenly the patient may die. That is why during blood transfusion, they have to check up the antibody and the blood grouping. There are further divisions of A positive, O positive, O negative, like that. But at present, you should know A group, B group, AB group, O group. The AB group has got no antibody. So the AB group has got no antibody. So you can donate this blood to anybody. I can give the AB group has no antibody. I can give the blood to the A group person, a B group person, or O group person. So AB is known as the universal donor. At the same time, O group has got both antibodies. No, I'm sorry. AB is a universal recipient. You can receive blood from the A group, B group, as well as the AB group. But there's no interaction. There's no already antibody there. If I have B antibody, I can't get A. If I have A, I cannot get B. This fellow has got nothing. So it is a universal recipient, receiver. In the case of O, you have got both the antibodies. So you can give them to A group or B group or AB group. So this is called a universal donor. So one of the questions they'll ask you, which is a universal recipient, which is a universal donor. What are the different blood grouping? In the system of blood grouping, it's only ABO grouping. And the groups of blood is grouped and according to the antigen. Antigen will produce antibody. The same blood is, if the different blood group is blood is transfused or administered, and the two different antibodies coming together will be dangerous and will club up the blood cells. You group the blood cells. Upper now, what is the definition for upper? This is called native antigen or inborn antigen. Inborn or native. Or it's also known as native antigen. Native antigen. What is the use of antigen? Producing antibody. Apart from that, anything what is the use of antibody? Producing resistance to your body, from immunity to the body, from the attack of the foreign bodies. So anything which will trigger the formation of antibody is known as an antigen. Say for example, apart from this, if I get an attack of a fever or a virus or a bacteria or a virus or a chemical which is not accepted by my body, and the body, suppose a bacteria comes, WBC goes and attacks them. WBC is a soldier of the body, and add on to that, these antibodies will be produced by the bacteria. Now, the antibodies produced for a particular infection. So if I have a fever of typhoid or viral infection, my body will produce, apart from the WBCs, which they are fighting against the virus which has entered into my body, these antibodies will be produced by the blood. What triggers the production of antibody? A bacteria or a virus or infection, pathogen. So these antibodies which are produced by the pathogen are not native antibody. When you get a fever and attack, these antibodies are produced and even they help to fight out the pathogen. At the end of the thing, you get over of the disease, the antibodies won't disappear, it will remain in your blood. That's what you mean by immunity. You are developing immunity or resistance against the disease after an attack. That is called active immunity. Now, so anything which triggers the production of antigen is an antibody is an antigen. Native antigen is A and B, and any infection pathogen can be an antigen, or sometimes a chemical which you can produce an antigen because your body has to protect itself, a self-defense mechanism. So anything which what is the antigen, anything which triggers the formation of the antibody is an antigen. So what's the use of antigen? Antigen is like a WBC like cell. It helps to attack. Suppose you get an attack of malaria or any disease. The WBC helps. That's the first line of defense in your body. And the first line of defense, it helps to, it tries to attack, destroy the pathogens which cause the disease. But the WBCs are not strong enough. The number of they are not able to kill the virus or pathogen. Pathogens are very strong and virulent, very strong in their attack. Your body, the WBCs are not able to control, your body is not able to stand the infection. In that case, antibodies will be produced. Now, these antibodies, how do they help? They help the WBC to attack the pathogen which has entered in your body. They'll club up all the virus, they'll group up all the virus so WBC can swallow that. During the interaction, the WBC and the pathogen, poisonous substances will be produced. So the poisonous substance will be nullified by this antibodies. So antibodies is a secondary help for the WBCs to protect your body. And not only that, they remain in your blood after you got cured of the infection. They'll remain in the blood for about they remain in your body even after you got cured of the disease, at least for 3 years it might remain. So when they remain for 3 years at least, you should not get the attack back again. That is, suppose you have an attack of typhoid or jaundice a year. Once you cure it, come out of it, next within at least within 3 years or 2 years minimum, you should not get the infection back. That means you haven't developed resistance. So the antibodies provide the resistance. Now, when you get resistance after an attack of a disease, it's called active resistance. So resistance or immunity, antibody produce immunity. That is active immunity. Active immunity means you produce resistance after you get an infection, then you develop this. And this should be remaining there at least for 2 years. Then suppose you don't want to get a fever. You don't want to undergo the problem, passive immunity. You don't want to undergo the pain of fever and everything. Passive immunity means what? They will produce antibody artificially, okay? And that will be introduced into our body. Or they can two ways. A passive immunity, active immunity, and passive immunity. Passive immunity is you don't want to undergo the fever. So what you do? Either you get two ways: vaccination or inoculation. Vaccination or inoculate. What do you mean by inoculate? Inoculation means they take the pathogen. In vaccination, the antibodies are produced. Antibodies are produced artificially. How will they produce antibodies? How will they produce antibodies artificially? They will take a specimen, that is invariably horse or a cow. They take the horse or a cow and inject the virus, the pathogen into it. So the body of the animal will react to that and it will undergo the fever and everything and it will produce antibody. Then they will separate the serum of the blood. What is serum of the blood? They'll take the plasma, remove all the fibrinogen and everything, and the liquid plasma without clotting factor is called serum. So the serum will have the antibodies. So then they will take the antibodies and inject into my body. So I'm already getting the antibody to resist against this. Is passive immunity through vaccination. So every vaccine has got a pre-prepared antibody. And the inoculation in a sense, they will take the animal, that is, they will dilute the pathogen, they will directly introduce into your body. So the pathogens are directly weakened pathogen. So it is called weak pathogen or weakened. One is vaccination, another one is inoculation. Another one is inoculation. Inoculation means in this they will give a weakened pathogen. Weakened pathogen. It made weak. Weakened virus or a bacteria and pathogen. Weakened pathogen. Pathogen or virus that will be introduced into my body and the body will respond, it will develop a fever. That is vaccination and inoculation. These are the two ways they inject in you. Either your pre-prepared antibody is sent into your body or a weak pathogen is sent and you develop the fever and develop the resistance. That is passive immunity and active immunity. Active immunity is, no, I'm sorry, once again. Immunity or resistance. Inborn immunity, inborn. Then acquired. Acquired means you get it outside. This can be active or passive. Passive immunity, pre-prepared, already pre-prepared. Prepared antibodies are introduced. In this active immunity, weak pathogen, weakened. Weakened means they made weak. Weakened or made weak. Weakened pathogen will be introduced. Pathogen is inoculated or vaccinated. The vaccine contains antibodies, inoculation normally is this. So the vaccination or inoculation, you call that as vaccine contains all the prepared progesterone or diluted, or another method is also known as inoculation. Inoculation. That is your acquired immunity, you don't have inborn, haven't got it by birth, you are getting it after you, when this is actually when you inoculate, what will happen? You develop fever and your body will react, you develop fever and then the body will produce antibody because you are injecting only a pathogen, is an antigen. So you will produce the pathogen is an antigen which triggers the formation of antibody. So the antibody, this kind of developing resistance after getting the fever is called the active immunity. When you don't want to undergo the fever, you develop the antibody pre-prepared and introduce in your body with vaccine. That is called passive immunity. So acquired immunity is of two types, active and passive. Active is fever, passive. In the case of inborn immunity, you are possessed by, this is by the blood group A antigen, A antibody, B antibody, AB antibodies. So the resistance, the antibodies produce mainly immunity or resistance against, and they are the second help of the WBC to attack your protect your body from the pathogen. Thirdly, they remain in the body for a few years to prevent the attack of the pathogen back again. Now this is about the blood grouping. Now the next one, there is another factor which is known as the RH factor, RH antigen. This RH antigen, antigen is nothing but a kind of protein. Now this RH antigen is found in the rhesus monkey in Africa, you know that. Now this is found in certain persons. The persons which has got this addition to other group, they have the RH antigen, they call RH positive. The person who does not have is the RH negative. So by nature, when these two cannot come together, because you know very well, the RH negative will produce antibodies against RH plus. RH plus. If the two blood comes in contact, then the interaction will be there. So now what happens? This case comes when the RH negative female having the marriage during a female having an RH negative blood comes in contact with the RH positive blood married to a RH positive man. Now another thing is RH positive is a negative, is a dominant gene. So the dominant gene can be expressed in a double dose or in a heterozygous or homozygous condition. So when the mother is a RH minus and the man is a RH positive and it develops the fetus develops. And the fetus being the developed fetus will have, because it has a RH positive. The fetus blood and the mother's blood are separated. It doesn't come in contact because the placenta, through the placenta, it is separated. The barrier tissue is there. But during delivery time, that is during delivery, this is one other question they'll ask you when they come in contact. During delivery, the positive blood of the child is all the positive blood are coming in contact with the negative blood. The mother's blood will come in contact with the positive child, the fetus. It's a fetus. Fetus when they marry together, that is RH negative female and RH positive RH positive male. The product is a RH positive fetus. Now, what will happen when the mother's blood comes in during delivery? During pregnancy, no reaction. Now, pregnancy they are separated by placenta because this is a question they'll ask you. Placenta, the RH minus individual marries an RH positive. Now, what will happen? Invariably, the blood, when the mother's blood comes in contact, the fetal blood will be plus, then only the reaction will be different. So, what will happen during delivery? The RH negative mother comes in contact with the RH positive of the fetus. During delivery is one of the questions. Then what will happen at that time? It will produce antibody against it. Antibody against the RH positive. So during the second conception, now these antibodies will pass through the blood of to the fetus and destroy the blood cells of the fetus, and the fetus might not develop or it will die. Now, invariably, in a book it will be given the child born is the jaundice and disorders. But normally, in certain cases, the survival is not possible at all. So that's called erythrocystic blastolysis. Erythrocystic blastolysis. Blastolysis means lysis means splitting. Blastula is one stage of development. Erythrocystic means due to the RBC, deformity in the RBCs or blood cells. Erythrocyte is RBC. Due to the deformity or destruction of RBC, the developmental stage itself, the embryo won't develop. So invariably, it will be a stillborn death. Second conception will be aborted. Blastula is the early stage before you come to the 12 weeks time. So the blastula stage itself, the zygote won't proceed beyond that. The fetus might die, or it will be aborted. Otherwise, sometimes if the child is born, depending on density, and it will be in the textbook, it will be given jaundice. It suffers with the jaundice and deformity. So this is what I mean by RH incompatibility. What is RH incompatibility? When an RH negative individual marries a RH positive individual, first conception will be escaped, the child will be alive. So these people invariably in this couple will have only one child will be there for them. That will be the first one. After that, the second and third conception will not be successful. Now, when does the mother? What are the questions here? They'll ask you what is the RH incompatibility. There are the antigens of RH plus and RH negative. They are not compatible. They are not adjustable. They cannot get along. In other words, incompatibility. Then when does the mother's blood comes in contact? During the delivery. So how to prevent this? They will inject during the delivery into the mother's blood the RH plus antigen before itself. That could be avoided. That could be prevented. This happening. So the RH fetus blood and comes in contact, RH negative mother's blood comes in contact with the fetus blood during the delivery. So the mother's blood begin to produce antibody against the positive, it is there in her blood. So during the second conception, that antibodies will not allow the zygote to develop, and invariably the embryo won't develop finally after the blastula stage, or even if it develops, the child born will have a defect, it won't survive long. That is about RH factor in this. The next one which you have to do is a blood clotting. Blood clotting. Clotting of the blood. Blood is a little connective tissue. How the blood becomes solidified. That's the word blood clotting. Now the blood clotting is a long process. Now in this first the clotting prevents bleeding. Prevents bleeding by closing the cut ends of the blood vessels. So blood vessels. So what happens? Cut injury. Injury or a cut. Now the tissues are as soon as the injured tissue produces injury and the injured or damaged tissue. Injured tissue produces a chemical, forms a chemical which will attracts platelets. Platelets are a kind of blood cells. They rush to the place of the injury. So now here the platelets rush into that injury. Okay, the platelets race. The platelets are triggered by the collagen fibers. The cut end of the fibers induces the platelets to produce two things. One is it produces serotonin. The platelets produce, that is stimulated by the tissue of the damaged tissue. The damaged tissue has got collagen fibers. Now the collagen fibers of the damaged tissue makes the platelets to produce two substances. One is serotonin. Another one is thrombokinase. Now the serotonin is a vasoconstrictor. So what will happen? It will narrow the cut blood vessel. The end of the blood vessel will come closer so that the blood cannot ooze out. At the same time, the blood flows out. The solidified clot will not be washed out by the blood. Serotonin is a vasoconstrictor, narrow the blood vessel. Automatically, the cut end is narrowed and the blood loss will be less. Now, the thrombokinase is a kind of enzyme. This will act on a substance inactive form called prothrombin in the blood. Prothrombin in the plasma. That is already in the plasma. And the prothrombin is produced by prothrombin is produced by a liver. Prothrombin is produced by the liver with the help of vitamin K. Vitamin K is produced by an inactive form. Prothrombin is converted into thrombin in the presence of, now one second. Thrombokinase acts in the presence of calcium ion on prothrombin. Prothrombin is an inactive form. It's produced by the liver. The liver needs vitamin K to produce that. So the prothrombin and thrombokinase combines in the presence of calcium and the prothrombin is converted into thrombin. This thrombin acts on in the blood or plasma you have got a soluble form. Plasma has a soluble or a liquid state fibrinogen. It's a protein. The soluble form, fibrinogen. That is converted into solid fibrin. Fibrin forms a network. In the network, the dead RBCs, WBC, blood cells, pathogens all get together and form a clot. The network gets trapped. So this forms a clot. The clot, already the blood vessel end is narrowed down, and the clot plugs the narrow part of the blood vessel and prevents the next bleeding. So the clot prevents the bleeding by this process. So the cells important for the clot formation is the platelets. Now add on to that, the platelets will be there, the cut injury, they keep on producing secrete the serotonin and the thrombokinase. Now these thrombokinase and serotonin will be at the end, they will polymerize. Polymerize means they destroy the muscle fibers near the wound. So near the wound, the muscle where the clot is formed, and again the platelets will produce the serotonin and thrombokinase. They will keep on producing these two substances, and surrounding the clot, it will make a elastic fiber-like structure that will cover the clot. So the clot will become very firm and thick. So that is called the clot will not remain, it will become hardened by the apparatus, contractile apparatus, like that will close up the clot and make it hard. So the injured tissue produces a substance to attract the platelets to the spot of injury. And as it reaches the spot of injury, the platelets produce two substances, that is serotonin and the thrombokinase. Serotonin is a vasoconstrictor, it narrows the blood vessel, cut into the blood vessel, so prevent the bleeding further. And the thrombokinase is an enzyme which, as in the presence of calcium, it converts a inactive form of prothrombin into thrombin. The prothrombin is produced by the liver in the presence of vitamin K. Vitamin K is essential for the production of prothrombin. And the prothrombin converts the thrombin. Thrombin in turn converts the plasma protein in a dissolved state. This is in the blood. Now this is in the, this is in the liver. This whole thing up till this is in the platelets. And this is in the plasma. Now this section is in the plasma. This whole thing you have got in the blood cell. Platelets. So platelets, blood cell. So this is a self-defense. This is an injured tissue. So injured tissue triggers the platelets, and platelets in turn combination with the liver. So the question can be now, listen. Now after this form of soluble form of in plasma, this is the plasma, into a thread-like structure, the fibrin is a solid elastic thread. Now these threads form a network. In the network, all the dissolved pathogen, RBC, WBC, the blood cells get trapped and form a lump. That lump is called clot. So already the blood vessel is narrowed and the lump close to the blood vessel. To make the clot firmer, now the platelets will still continue. It will split the actin and myosin of the muscle near the wounded area. And actin and myosin of the muscle of the near the wound gets polarized, okay, then polymerized, form a thick covering over the clot. So the clot becomes firm, closing the blood vessel. Now this is a process of clotting. Now, how the question will be in the antigen and blood grouping and this. The question they can ask you, what are the different antigen and antibody? What is antibody? How it can be, it can produce resistance. So they can argue choose the correct answer. Antibody provides resistance, carries oxygen. It's a defense mechanism. Actually, providing resistance is the major work of antibody. They can give you universal donor is A, B, or O. So universal recipient, choose direct answer questions will be there. Then RH incompatibility, the marriage between RH negative female and positive male. RH negative male and positive, they can change over. If the female is negative, male is a positive individual. Then RH, the thing, now why the erythrocyst, the condition by RH incompatibility of RH factor is called particularly since this where is They'll give you RBC, bone marrow, blood, liver. Or essential for vitamin K is essential. Now, in this disease, a disease where the clotting does not occur, is a genetic disorder which you already known as hemophilia. Hemophilia, hemophilia. It's a genetic disorder. It's also known as bleeder's disease. Bleeder's disease. Or it is also known as royal disease. Hemophilia is hemophilia or it's a genetic disorder. This is the gene, hemophilia gene, and the mothers will be the carriers, females will be the carriers, and the males will be the affected ones. Now, the hemophilia, it is known as the bleeder's disease because the bleeding clot formation capacity will not be there. The reason can be the lack of vitamin, lack of any one of the factor. Particularly, vitamin K deficiency also will help in the hemophilia. Glad on. If you don't have enough K, prothrombin will not be produced in the liver. Prothrombin only will form the network, finally forms the fibrin, leading to the formation of prothrombin will be converted to thrombin. Thrombin only converts fibrinogen into fibrin, then only clot formation. And it's called royal disease, as you know, it was first found in the royal lineage of Queen Victoria. So the hemophilic disease is a disease formed due to the lack of blood clotting. Okay, now in this, the next question can be invariably they'll ask you, where is the thrombin produced? Prothrombin is produced, or options will be given. Now, liver and calcium ion, or liver and vitamin K. This vitamin K, not the ion, the vitamin VK. And another question can be in the calcium. What is the necessity of calcium in blood clotting? Calcium helps in the action of thrombokinase. Another word for thrombokinase is prothrombokinase. Because it's converting prothrombin, it is also known as thrombokinase or prothrombokinase. Prothrombinase it is known as. So prothrombinase means convert the prothrombin to thrombin. Okay? So they'll ask another question oriented to the calcium. The ion needed for the formation of clot, they'll give you. And that clot is calcium ion. Sodium, potassium, anything, calcium is important. And similarly, the calcium is necessary for the contraction of the muscle contraction. So the calcium ion in the clotting is the chemical ion mineral needed is calcium. Another question they can ask is soluble protein in the plasma, which helps in clotting is the fibrinogen. Fibrinogen is soluble in the liquid of the blood. It will be converted into fiber clot. Clot contains. Next question, clot contains the network of the fibrin network you should have with the dead RBCs and everything, and is reinforced or made it hard with the help of the actin and myosin fibers around the wound, which is depolarized by the depolymerized by the platelets back again. The chemicals produced by the platelets. So these are the questions related to your clotting and blood grouping. The next one you have to do is the control of the cardiovascular system. Before you go to the control, the function of the blood function of the blood. One of the major function of the blood, which you should have done it in the last class. It's the transport of respiratory O2 and CO2. This is one of the major function of the blood. Now, in the case of transport of O2, (97% by hemoglobin (Hb), 3% by plasma. Hemoglobin is a pigment in the RBC, and the RBC hemoglobin is a Hb or hemoglobin. It's a pigment, coloring substance in RBC. And you should know, RBC is a cell, has no organelle. Matured RBC has no organelle. So it cannot reproduce. It It can't reproduce. Neither it can breathe. No mitochondria, no cell organelle, anything. And it has got only the pigment heme plus globin. Heme plus globulin is a protein. Globulin. So one atom of Hb, molecule of Hb, one molecule of Hb, 1 mole of Hb, that is Hb4, carries 4 atom of oxygen. So the amount 100 ml of oxygenated blood can give you 5 ml of oxygen. So the oxygenated blood means what? When the Hb combines with the oxygen, it's called oxygenated Hb, oxyhemoglobin. And another property, the hemoglobin has got the capacity, tendency to combine and dissociate quickly with O2 and CO2. It can combine with O2, it can combine with CO2 also. And it will dissociate from it, leave out O2 and leave out CO2 easily. So it has a capacity. The next capacity is carries 4 atoms. This is one of the questions they can ask you. How many atoms of hemoglobin molecule can carry? It can carry 4 atoms of oxygen. And then it can easily combine, associate and dissociate. Associate and dissociate. Dissociate. Dissociate with oxygen. Okay. So these are the peculiar property of the hemoglobin. Okay, now the hemoglobin is present in the RBC maximum, the pigment. The RBC has got nothing but a body of the RBC has got a network of fiber. On this fiber, the hemoglobin is trapped. All these are the hemoglobin on the fiber, trapped on that. That's all the structure of this RBC. HB, hemoglobin, combines with the oxygen, maximum 97% of the oxygen is transported in the blood by the hemoglobin and 3% by plasma. But when you talk about the transport of CO2, that is 20 to 25% by RBC. It will become carboxyhemoglobin. Then 3 percentage, 25 plus 5, 3 to 5. Say 20 to 5, 3 to 5 percentage will be by the plasma. Dissolved in the plasma. Dissolved in the plasma. About 70 percentage. About only, not approximately. It is transported as bicarbonate salt. This bicarbonate salt can enter into the RBC or in the plasma. It has got chemical reaction. So there are 3 things. The bicarbonate salt involves in the transport of CO2. It doesn't come in the transport of O2. So this percentage you should know in your Neet paper. What are the percentage of what are the 2 mode of transport in oxygen in the blood through Hb and plasma. No salt involved. And which is the ion involved in the transport of CO2? Bicarbonate ion. And which is the maximum amount is carried by the of CO2 is carried by the salt, by not in the Hb or a plasma. So these 3 points you should remember when you comparing to this. Okay, now you know very well the transport of oxygen, these 2 gases are exchanged. Now where are they exchanged? Now these 2 gases are exchanged. Exchanged in 2 places in your respiratory system. That is, oxygen is loaded. Loaded means it combines, it is received in the lung. How? In the lung? By the blood. And it is deposited. It is loaded. Oxygen is loaded or carried or gets into the blood in the lungs, and it is deposited or delivered. Delivered or deposited in the tissue. So oxygen from outside, from the air. Here comes your respiratory system, alveoli, exchange and everything. Gets into the blood is purified, and the purified blood delivers it to the tissue. Now this is O2, direction. In the case of CO2, The production of CO2 is produced in the tissue because formed in the tissue, produced in the tissue by metabolic reaction. So the loading of CO2 from the tissue, in the tissue, loading. And where does it deposited? Finally, it carried via blood. It is sent to the lungs. So oxygen from lung to the tissue, CO2 from tissue to the lung, and from the lung it is expelled out. It goes out. It's coming in. Alright. Now this exchange depends on three factors. That is, it depends on partial pressure, this exchange depends on four factors. The exchange of O2 and CO2 depends on number one, partial pressure PO2, PCO2, then hydrogen ion concentration and temperature. Invariably, the PCO2 and temperature will go together. If PCO2 is more, hydrogen ion will be more, temperature will be more. And though these four factors help in the exchange. What do you mean by that? If the PCO2 is more in the gas and O2 pressure is less in the tissue, so the O2 is from the blood, it goes to the tissue. That is, concentration of PCO2 is more here and it is less here. Say for example, PO2 is more. Now here PO2 is less. So the transfer comes over here. But in the tissue, CO2 is more. CO2 is more, and here in the blood, the PCO2 is less. So the flow is from here to here. So depending on the partial pressure of these gases, the exchange of gases occur immaterial from blood to the tissue or tissue to the blood. Via blood, it is expelled out. And a book will be given exchange in the lung. Loading is in lung, delivered in tissue. Loading in the tissue, delivered into the lung to be expelled. Now, in this, though there are four factors concerned, the PCO2 is more important than PO2. Now, when will the PO2 will diffuse into the tissue if the oxygen is less and CO2 is more? Then only the exchange will occur. So the transport depends mainly on, they will give you the choice. Now the exchange of gases depend on PCO2, CO2, H and methane. Which is the most important factor? Options PCO2, PO2, hydrogen, temperature. But there is the most important factor, the PCO2. Because of the pressure difference between the tissue and the blood, or blood and the tissue, arterial blood and the tissue, or venous blood and the tissue, the exchange of the diffusion of the gases occur. So PCO2, it does not occur. If PCO2 formation is not there, the breathing will not be there. The individual will be choked to death. So now, during the swimming time, people who go underwater and they die, the main things when they underwater they go, they breathe in and all the ATP is totally used up. That is, oxygen is used to produce energy and they have to take the next breath. When they take the next breath, only next oxygen will come. During the breakdown, CO2 accumulates in the blood, more CO2 in the blood, and they have to take a breath to expel out. If they are underwater, they are not able to continue the breathing. So PCO2 increases in the blood and lead to sudden death. That's a cause for the death of the people who immerse under the water. And the training people, that's why the swimmers, they have to train how to breathe out at that time. You follow? So the PCO2 is more essential of all the factors than the other thing. And another point is, the CO2 concentration will always go along with the H plus and temperature. If CO2 increases in the tissue, the temperature of the tissue and the hydrogen ion concentration is more. If the PCO2 decreases here, the whole thing will be reversed. So all the four factors, the CO2 will go along with hydrogen ion and temperature, but this will be opposite to each other. O2 concentration and CO2 concentration. Now this exchange of gases only, now this is the major point you should know in the exchange of how this is actually the blood, the blood is the main function, the oxygenated, deoxygenated blood, and the partial pressure of the gases dissolved in that only which causes all this. So how the O2 and CO2 is carried in the blood, those are the two different ways. Oxygen is 2 method, carbon dioxide is by 3 method. By salt also. But oxygen transport, salt won't come. Now this combination of oxygen with the hemoglobin, so oxygen combines with HP. Now this is shown in a graph. That is a graph, it's called dissociation graph. In the dissociation graph, partial pressure of oxygen will be there. This is part PO2, partial pressure of O2. Now here, saturation of HB, saturation of HB. Saturation of, 1 minute. Saturation of HB occurs. So in this again you have take 20, 10, 20, 30, 40, 50, 60, 70, it goes on 70, 80, 90, 100. Now let it be 10, 20, 30, 40 like that. This is partial pressure of O2, saturation level of HB. Now that gives a sigmoid curve. The curve is called sigmoid curve. In the early stage it's a steep, that is 10, 20, 30. Up till that, the oscillation will be very sharp. Then gradually it gets a slope. Maximum saturation is the 70 mm. At the level of when the partial when the maximum saturation occurs, that is in this peak, at 20 by the time I have to draw the thing properly. 10, 20, 30, 40, 30, 40, 50, 60, 70. Let it be 70. So at the level of 70, maximum saturation will be there. It will not be 100%. It can be 80 or 80 to 100, but not 100. So this maximum saturation is between 80 to 100, and at the partial pressure of 70 mm Hg. See the maximum, there is no 100% saturation in the case of HB, and the curve is a sigmoid curve. Now this is the sigmoid curve. Now if you take a thing, now sigmoid curve is there. Now this is, we are talking about PO2 and HB here, saturation level. Okay. Now this, we suppose I told you what, the partial pressure of oxygen is not very important. PCO2 is more important. So if the PCO2 increases, now what will happen? So the combination with the HB will decrease. Oxygen capacity combining will be lesser. So what will happen in the P? Normally it is at this time at the maximum normal curve. The CO2 is 40. We saw in the tabular column in the respiratory system. If the CO2 increases, the graph will come down. That is 60 PCO2. That it will shift to the, this is called right shift, or it will come below if the CO2 increases. The CO2 decreases, so 3 PCO2. The graph will go up. This is called left shift. This is a normal curve. Normal. This is a normal curve. Normal sigmoid curve. So this is called dissociation graph. Oxyhemoglobin oxygen dissociation graph. When the oxygen will be the maximum saturation of HB and how it will release the HB. Now release of oxygen from the HB depends on the partial pressure of oxygen. So when the CO2 pressure increases, the graph will shift towards below. It is called right shift. When the CO2 level decreases, the graph will move up. It is called left shift. Now this is called Bose effect. Bose effect. Also, it is also known as Hamburger's effect. Hamburger. That is the oxygen dissociation graph, a sigmoid graph. When it changes the position up or down, if it goes up, it's called left shift. If it comes down, it's a right shift. When will it go up? The 30 when the CO2 is lesser. When CO2 is lesser, more oxygen will be getting into the HB. So the oxygen combination will go up. So the CO2 will become lesser. If the CO2 increases, the oxygen combining capacity, saturation of the HB will be lesser because it will be saturated with the CO2 as well. So this is this graph you should know. This is one of the major function of the blood. Transport of respiratory gases, oxygen and CO2. Which form it is transported? In the RBC, oxygen is through RBC and plasma, and CO2 is through RBC, very little, 25%, 20 to 25, 7%, 20 to 23, 7% by the plasma, 30. The remaining 70 is mainly by bicarbonate salt. Now then for the where it is loaded and oxygen is loaded in the lungs, delivered in the tissue. And CO2 is loaded in the tissue, delivered in the lungs via blood and to be eliminated. So for this exchange and loading and unloading depends on 4 factors: PO2, PCO2, hydrogen ion concentration and temperature. And PCO2 is the most important factor than the other 3 factors. So to show what is the partial pressure of oxygen against the saturation of the Hb, you have a curve that called oxygen dissociation curve. That is, it is maximum saturated. It has to give out oxygen. But when it has to give out oxygen, it depends on the partial pressure of CO2. Now, if the CO2 is more, then oxygen, to remove the CO2, oxygen has to go in. That's the meaning. So now CO2 pressure, the curve is a normal curve sigma. That one question will be there. Now this itself, they can ask you so many one-word questions. That is, the maximum, when does the maximum saturation occurs at mmHg? And there is no 100% saturation. The maximum is 80 to 90, 80 to 90 only. Now 70, 80 to 85 or 90. Then what is the shape of the curve, sigmoid curve? And the beginning stage of when the saturation is the early stages, that is when the pressure is 10, 20, 30, the saturation is faster. Then the saturation will slow down and become a flat when the PO2 increases. Now the maximum concentration is saturation is by 70 mmHg. The partial pressure of oxygen. We saw partial pressure of oxygen, the tissue is 95 mmHg, the blood. The tissue will be less. So now when the curve, if the CO2 pressure, partial pressure increases or decreases, now this curve will not be a sigmoid curve, it will shift downward or upward. When the CO2 pressure increases, it will shift down, you call that the right shift. When the CO2 level decreases, it move up, that's called the left shift. Now this shift is known as the Bose effect or Hamburger's effect. Now this is a function of the blood in transporting O2 and CO2. The next one we are going to see is the control of function of the cardiac function. Control of cardiac function. See, if you take the heart, you have the heart. Heart. Now you have SA node. Okay. Now this SA node, cardiac control. Control of heart function. Now if you take the heart now, you have structure of the heart. What I meant is the ventricle, atria, ventricle, and you have SA node. You know, your heart is myogenic. Why is it called myogenic? The stimulation of the heart, the functioning of the heart, the depolarization occurs at the SA node. And SA node is a myogenic, it's a special muscular self-excitable. That is, there is no need for any supply of the nerves or anything to trigger the reaction. Automatically, when the atria is getting filled up, or even the filling up of the atria might not be, just self-excitable. That is why it's called myogenic heart. Myogenic heart. It's not nerves coming and stimulating. Myogenic heart. And when the SA node, so if the SA node triggered only, the contraction will start. And totally it is under ANS, autonomic nervous system. In autonomic nervous system, we have got sympathetic and parasympathetic and sympathetic fiber. Sympathetic. Now you have got, there are two fibers, both the fibers are supplied to the SA node. This is SA node. SA node receives parasympathetic fiber and sympathetic fiber from the brain proper. So let us take this is a brain proper. This is sympathetic fiber. Both are coming down to the sympathetic as well as parasympathetic comes to SA node. Parasympathetic. Comes to SA node, comes down from the brain. Now, so when they come to the SA node, The stimuli, which is when the contraction of the SA node will spread. A sympathetic fiber slows down the contraction, and parasympathetic fiber accelerates, speeds up the SA node, acceleration. It's an accelerator. It speeds up the spread of the stimuli and contraction. It slows down the contraction. And this parasympathetic fiber passes to the 10th cranial nerve called vagus. Because these sympathetic and parasympathetic fiber find their route to the organ through the cranial nerves. Some will be through the 9th cranial nerve, some will be through the 10th cranial nerve. The cranial nerve is a carrier of these fibers. There are only fibers, parasympathetic, sympathetic fibers arising in the particular region of the brain tissue. And they come out other along with some nerve. They get mix along the nerve, and then, but these are only fibers. So the nerves are nothing but bundle of fibers. Some of the fibers are, when parasympathetic, sympathetic are opposite to each other in their function. If one increases the speed, another will decrease. And through which nerve it comes parasympathetic completely to the 10th nerve. Now both of them will reach the SA node, and SA node is auto-excitable, that is depolarized, stimulated. The stimuli and that this will enforces the stimuli, and the stimuli spreads all over here. Now this is the ANS. Apart from that, you have got in the heart, if you take in the the structure of the heart, you have got other important area of the heart where you have the arch of aorta. The arch of aorta from the left V. And from there branches, if you remember, there are 3 branches. The common carotid, this is the left common carotid, LCC, left common carotid. Now this is the left subclavian. Now this is the common subclavian carotid. It branches into 2. Now this is the right common carotid. Right common carotid, that will divide into 2, internal and external. This common will go up and divide into 2. Will go up and divide into 2. This is a left common carotid, goes inside, divides into internal carotid and external carotid. At the base of the carotid artery, you have got an area called carotid sinus. Carotid sinus. At the same time, on the arch of aorta itself, there are aortic sinus. Aortic sinus. Now, when these cells, that is, state stimuli, when the blood stage passes out during the contraction, blood is pumped out. So automatically it will go pumping through the blood vessel. It will stretch stimuli. This blood vessel with the same force it has to go up. So at that time, these cells on the wall of this carotid, internal carotid, where both of them divide, that's called carotid sinus. This is the aortic sinus. The aortic sinus and carotid sinus are stimulated. Now, when they stimulate, what will happen? They will increase or decrease the heartbeat. So they will slow down the heartbeat. Now, simultaneously, you have got sinus venosus and the superior vena cava. Superior vena cava and falls into the superior vena cava falls into the, very well, right atrium, and inferior vena cava also into the right atrium. So the superior SVC at the base, you have got the sensory cells. That is the receptors of the SVC. That is the coming in blood. Now this is going up blood. So this will be these things will stimulate and increase the heartbeat. So SVC, that is superior vena cava, the sinuses of the superior vena cava, receptors of superior vena cava, receptors increases the heartbeat. Increases the heartbeat. Aortic sinus and carotid sinus and aortic sinuses decreases the heartbeat. Decreases the heartbeat. So the rhythm of the heartbeat decreases the beat. So the contraction or spread of stimuli from the SA node or triggering of the stimuli along with the myogenic center. SA node is a myogenic center. So specialized self-stimulated muscle tissue. When that stimulation is strengthened or enforced so that it can spread out throughout the heart with the help of the ANS. So ANS and the ANS system is autonomic nervous system. It has got two fibers, sympathetic and parasympathetic. Now sympathetic and parasympathetic arises from the medulla. Now where are these? Now these are sensory fibers. From here the sensation will go up to the medulla oblongata. That is from SVC. Now wait one second. So now if you take now superior vena cava and carotid sinus, aortic arch, receptors in the aortic arch. So these two things will decrease the heartbeat, and this will increase the heartbeat. That is, these fibers are actually, it will carry the message, both of them will carry the message to medulla oblongata. The medulla. The medulla, you have a cardiovascular center, medulla oblongata. This is the lowermost part of the brain. The medulla oblongata, you have cardiovascular centers. Now these centers receive the stimuli. Now when the blood vessels dilate, SVC will dilation of the blood vessel, that sensation will be carried to the medulla oblongata. In response, the response will be in a different manner. It will increase, the responses will cause a dilatation. The stimuli from the SVC will reach the medulla oblongata, which will dilate the blood vessel, and the blood flow will be more. And the receptors of the cardiovascular, the aortic arch and carotid arch, receive the sensation and send to the medulla oblongata. And the response make the blood vessel contract. So the response has the effect. So the sensation, the sensory area where the sensation goes to the medulla oblongata. Response comes down from there. But ANS in turn controls the through the SA node. There's a rhythm of the heart. This is. So the systole diastole. Systole and diastole is controlled by the SA node through the ANS. But the volume of the blood, contraction of the blood vessel, dilatation of the blood vessel is controlled by the receptors in the sinus vena, aortic arch and carotid sinus, as well as vena cava, superior vena cava. Both are different. See, the ANS controls the working of the SA node. That the stimulation is set on the SA node, then only the diastole and systole will start automatically, cardiac cycle. But this one, the sinus venosus and carotid arch, when they are receptors, these receptors send the message to the medulla oblongata, which in turn will dilate the blood vessel or constrict the blood vessel, blood volume. So aortic arch, carotid sinus, and the superior vena cava regulate the volume of the blood. Now the SA node, ANS, via parasympathetic, sympathetic, controls the working of the systole and diastole of the heart. So systole and diastole, there is rhythm of the heart, increase and the amount of the blood flow through the blood vessel, both are controlled by this route. ANS control the systole and diastole, and the receptors in the SC, the carotid artery, aortic sinus, and SEC regulate the size of the blood vessel, increasing the blood vessel dilates, volume of the blood will be more. You follow? So these are the two ways. Two are in different manner. The cardiac function is controlled. Now the next one is you have to see the cardiac defects. One is CAD, coronary artery diseases. That is coronary artery diseases. There are two types which I already told you, arteriosclerosis and atherosclerosis. Now this is by the deposition of calcium to the deposition of lipid. So both the cases, the arteries, the lumen of the artery will form the plaque. Suppose this is a blood vessel. Now the deposition of this will form a small mound that is called plaque. Now the plaque, this is a blood vessel, capillary particularly. Now the blood vessel, the plaque will increase by the deposition of more dead cells on them. So it will become a small mount. So the space inside will become narrow. So the amount of blood carried will be lost, and in the case of calcium, the blood tube will become more brittle, easy to break down. So that is CAD. Another one is the heart attack. Heart arrest is different. Heart failure. Heart failure in a sense, the blood supply to the heart muscle is insufficient. So the heart pumping action of the heart gradually fades. That is heart failure. Heart attack means the attack in a sense, the heart refuses to beat. That is heart attack. Now the heart failure is different, heart attack is different. Now that is why it begins. It can be sudden dead tissue, formation of the some of the tissue, but the lack of oxygen and blood supply, the tissue dies, and that is heart attack or heart arrest. Okay, heart failure is insufficient supply of oxygen or blood to the heart muscle, so gradually the working degenerates. So you have to be careful when you have the patients. Another one is angina. Angina means severe heart attack due to the infarction of the tissue. Infarction. Now, infarction means dead tissue. That is, heart muscle tissue dies gradually. It cannot occur all of a sudden once. Now, gradually, why it dies? Because again, insufficient supply of oxygen or blood. Blood, oxygen. So, the infarction of the heart, this is a gradually the thing, degenerating disease. It's not an all of a sudden, it might not, angina will come. The individual patient will suffer with the severe chest pain and referred pain on the right shoulder and right arm. That is called angina pectoris. Pectoris in the sense, this is the pectoral muscle. Flight muscles are the arm muscle, chest muscles are the flight muscle. So there will be a spread severe chest pain and referred pain along the right arm and the back of the shoulder. Now that is angina. So these are the certain terms related to the heart cardiovascular disorder. Now this finishes your vascular system. Regulation of the respiratory centers. See now this is a blood respiratory system. Next we have to go to the excretory system. That's okay. I finish the cardiovascular system. Should I continue or just stop now? That is okay. That I told you. Now we can see the overall. See, you know very well the questions which I already told you can be objective in this topic. Now the objective, it can be match the following, or it can be multiple choice or correct statement. Okay. Another important is the respiratory center. Respiration is controlled by four things. One is rhythmic center, pneumocognitive center, and the chemosensitive center. See, because respiration and blood vessel, we saw the control of the heart. Now, how the respiration is controlled? Respiratory control. Now, in the entire chapter, cardiovascular chapter, I have already discussed piecemeal. Now, what blood clotting, we can have a lot of questions. And control, what question can they ask the cardiovascular control? Now, the heart diseases, now before I go to the heart, we'll finish this and then you'll know. Respiratory control, one, respiration is controlled by four, the brain proper. One is rhythmic center. Rhythmic center. The rhythmic center is in the medulla oblongata. Medulla of the brain, it sets up the rhythm. How many times? 12 to 16 times a person can respire per minute, the normal rate. Now this rhythm is set by the in the medulla, center in the medulla. This rhythm in turn is affected by another center is called pneumocognitive center. Now this is in the pons. Now this pneumocognitive center in the pons and pons regulate the rhythmic center. Now the rhythmic center is back again. There is chemosensitive center. Chemosensitive center. Sensitive. Chemosensitive center. Sensitive center. Now this center is sensitive to response to the chemical. Suppose the CO2 accumulation is more in your blood. It responds to the chemicals which are getting into the blood. Now that in turn back again affects the rhythmic center. Suppose you need rhythmic center means what? Inspiration, respiratory rate. Suppose you need more oxygen, the rate of respiration will increase or decrease. So the chemosensitive center again sets up the rhythmic center. So the rhythmic center in turn controlled by the pneumocognic center, which is in the pons, and the chemosensitive center back again in the brain proper. And these things sets up the regulation of the rhythmic center. Add on to that carotid sinus, carotid sinus, sinus, and aortic arches. The receptors in aortic arches and carotid center are back again. It is controlling the rhythmic center is under the control of pneumocognic center, chemosensitive center, as well as the sensory areas in the carotid sinus and the aortic arch. That is when the carotid sinus narrows down. Now this back again you will study in the kidney. When the amount of the blood flow decreases, it will be stimulating the center in the hypothalamus. That will stimulate the rhythm, and then the respiratory rate will increase. So a simple example, say for example, when you are playing, no more carbon dioxide is utilized when you are playing an athlete. More energy is produced. CO2 accumulation becomes more. So your lactic acid, anaerobic breaking down of the glucose occurs. Energy is released from the anaerobic breaking down of glucose. As a result, lactic acid accumulates in your muscle. So the muscle fails to contract. That's why you get tired. Now, when the muscle gets tired out, you are not able to run. What do? You fall down, you start panting faster. The respiratory rhythm increases. Then the respiratory rhythm, and that is what accumulation of the chemical, chemosensitive. When the accumulation of lactic acid is more in your blood, that your respiratory center rhythm is set, rhythm is increased, so take in more oxygen and oxidize the lactic acid, and so back again your muscle fiber comes to the original. So these are the different ways your rhythmic center is attacked and altered. Suppose the carotid sinus narrows down. Now, when the narrowing of the blood vessel, the volume of the blood will decrease or increase. We saw again. Now, the increase in volume of the level will cause your thirst stimulation of the thirst. Now, there is less blood flow, water accumulating in your body, or you excrete more of urine. Amount of body gets dehydrated. Now, when the body gets dehydrated, it stimulate in the brain. So back again, the sinus, the stimuli of the carotid sinus are enlarging and decreasing can also affect the rhythmic center. So the rhythmic center is the major respiratory control, sets up the rhythm. The rhythm is 12 to 16 per minute. It is under the control of pneumocognic center and chemosensitive center and the stimulants present in the carotid sinus and aortic arches. So this is completely about your respiratory system. So now, oxyhemoglobin, what are the multiple tests? Say, for example, choose the correct answer. Bow shift. Now, the shifting of the shape of the oxyhemoglobin dissociation graph is daily L shaped, pyramid shape, or sigmoid shape. So, first question can be shape of the. Now, we'll stick on from your blood grouping onwards. Now, RH incompatibility will start. RH incompatibility. Now, incompatibility. Choose the correct answer. They will change A, B, C, D. The correct answer is RH negative female with RH positive male. Then they will ask you. Now, the RH erythrocystis blastolysis. Now, the next one, erythrocystis blastolysis, or a fetus complained of jaundice and repeated diarrhea. It shows, say for example, a newborn fetus, a fetus. Fetus means fetus or a baby embryo. Baby will take as instead of the fetus, you can take it as a baby. So, newborn baby, the newborn. A newborn means just newborn baby, shows the symptoms of jaundice and diarrhea. diarrhea. Cause can be, one of the cause can be. First, they will give it because it's a newborn. So the mother is infected with the disease. Mother infected with the disease. With the disease. Or the baby infected with the disease. Baby infected with the disease. Or RH incompatibility. RH incompatibility in the parents. Incompatibility in parents. And option they can give you none. Or invariably this is one to muddle up. None or they'll say all. Both the ways they can trick you. There is none of the above, only one. The statement is this. They can ask you to choose the correct statement, or they may give you an incorrect statement. So the statement questions only you have to be very careful. This is in the erythrocystis blastolysis. Even the term erythrocystis blastolysis is a condition of RH incompatibility or the death of the fetus or the death of the fetus, or they can ask you the second delivery. They can muddle up everything, choose the correct answer. So erythrocystis blastolysis, positive character, how many statements are true? So RH incompatibility. The fetus is invariably the RBCs of the fetus will be affected. That's true. Then the second birth, that's the second child. The first child will escape the death or the infection because antibodies will not be produced during the during the delivery of the first baby only. The blood comes in contact, or which minus comes in contact with the plus, and antigens are produced against the plus. Till then, the baby will not be receiving any of this reaction. So the second born, or that is also correct. And one more thing, it is the only the first child will be there. The couple will have only one kid. All the four statements are correct. So they will ask you how many statements are correct, or they can give you only two statements correct and two statements wrong. Pick up the correct statements. So the statement regarding the facts, they can now orient the statement question. Now, when you go to the and blood grouping, in the blood grouping, they can ask you anything. Universal donor is in ABO type of blood group. Now, see, in ABO type of blood group, ABO, that is our system is known as ABO system. In ABO, female or a male with the A group. I'll tell you next one. A group possesses antigen, or they can simply twist the question. A man's blood having A group blood group can be administered. Suppose I have A group, antigen will be B. A group can be transfused, can be given or transfused to the person with the blood group, BG or blood group. Option A 1A, or option 2 B, A group blood or B group blood. Option 3 AB. The A group will have what? Antigen will be B antigen. So it can be given to which of the answers correct? It can be given to A, as well as AB group has no antigen. So it can be received to AB blood has got no antigen, antibody, this has got both antibody. So you can give to this blood group. A group can be given to or AB group. Similarly, B group can be given to B and AB. Because O group no antibody. So AB group has got both the antigens, no antibodies. Sorry, the O group has no antigen, has both the antibodies. Has both antibodies. No antigen. But this has got no antibody at all. Because it has got both the antigens. So transfusion of the blood to A group, because it has got no antibody, they still have a B antibody. It can be given to the A group because it's the same group. This person will have B antibody. And AB group, it can cover, it has got both antibody, no antibody there, this antibody can survive. If I administer here, what will happen? These antibodies will click. It has got both the antibodies. So the B antibody will click with the to opposite antibody. Similarly, if there is B group, it can be given B as well as A. So they can muddle up giving like this way. Or they can ask you one more question in a dramatic manner. A person met with an accident, and a person met with an accident, there's a profuse bleeding. In accident, there's a accident, met with an accident, and profuse, plenty of bleeding. Profuse means plenty, plenty of bleeding. And a person's blood is immediately administered. A person donated the blood. And what could be the donor, plenty of things, what could be the donor's blood? They didn't give you the blood group of this person, neither you have to check the person donated blood, which is given to the person who is suffering the accident. So what is the blood group? So the blood group of the person, the blood group of donor can be A, B, AB or O. So who's the universe? You don't know the blood group of the individual who was met with an accident. We haven't given whether it's A group, B group or AB group or O group. But this person's blood group should be able to give in any blood group. So that should be an universal donor. So they will give you a choice. One group, the blood group of the donor can be A or B or AB or AB or O or O. So what could be, who will be the universal donor without any antibody? Okay, so the blood group of the donor should be an universal donor. Which blood group, universal blood group donor, that we have to write. Sure, the question is very simple, and but the way they pose it will be different. So blood group, they can group it, they can ask you the question in any manner. What you have to remember, who's a universal donor, who's a universal recipient. Again, the blood group is controlled by multiple alleles. Now IA and IB and IO. So multiple allelism is a blood group. Blood group is multiple allele. Multiple alleles. That means it has got more than. That is IA, IB, IA, IB and IO. 3 genes control that. If the individual has got this capital A, capital B. That is codominance. Another one is a codominance. This is your codominance. That is A blood group. The genotype of A can be IA and IA. This is also A group. If IO is a recessive one. So if the individual has got IA and YO, both the ways he will have the A group. Similarly, B group will be IB and B or homozygous and heterozygous condition. Okay, and that is when in the case of IA and AB, both the genes are there and there is only one genotype. This both can express the character. That's why you get the AB group. So out of the 3 genes, now the 2 genes will exhibit AB group. Both the genes are able to express their character. That's what we mean by codominance. And it is controlled by 3 genes or multiple allelism. Remember that. A group can have the genotype IA and IA or IA and O. O is recessive to the other. Recessive gene is considered as O. And AB express both of them when they are in the condition of A and B. So now the blood grouping, one word they can ask you, is a multiple allelism or how many genes are controlling the blood group or why is it codominance? Now that it is as our biology, so this question can blood grouping should go along with the codominance and multiple allelism. Remember that. Now in the blood group, R which factor certain question, the blood group that one. In this one, what is the thing? Control. ANS controls, control. When you talk about the control of the cardiac function, now what question can I word? So I can give you, SA node is controlled by, controlled or regulated by, A, CNS, B, peripheral nervous system, C, ANS, autonomic nervous system, and B, self stimulator. No, none of the all, or all the above, all. So it is mainly through the autonomous nervous system or parasympathetic and sympathetic via the ninth nerve, cranial nerve. But actually, the main control is by the, though 10th cranial nerve is involved, you should not come CNS. CNS represents, now this is peripheral nervous system. This is completely nerves. CNS represents mainly brain. But the fibers are, though the autonomous nerve fiber starts in the brain, the fibers are parasympathetic, sympathetic only, control, it reaches the SA node. So the controlling of the rhythm is different, controlling the blood volume is different. So blood volume is controlled from by the sinus venosus and carotid artery, carotid sinus and archa aorta. Senses receptors in the archa aorta, carotid sinus and the superior vena cava. That increases or decreases the blood vessel and blood volume changes. But heartbeat rhythm is set by the SA. So there they can ask you. Systole and diastole is mainly under the control of nodal tissue, which is under the control of what it is. Blood volume is mainly controlled by the receptors on the blood vessels, which are there. That which will stimulate, which will decrease. So these points you have to be very careful. Okay. Then what is angina? Yes. Oh. Now angina pectoris. They will ask you the angina, severe chest pain. A patient is reporting of severe chest pain and referred pain on the right arm and the jaw. It represents heart attack, heart failure, or infarction, dead tissue in the heart, or angina pectoris. Angina pectoris is the name as well as chest pain and arm connected. So these are the different ways the questions can be ordered. You should know the main points in all this. So that completes your circular system. Next, you have to go to the S3. I'll see you in the next class. Thank you.